Now I am going to discuss about clinical point clinical points in neurons. In the neurons class, I have discussed about axonal transport. And this axonal transport is of two types, anterograde and retrograde, that is forward and backward transport. So, in the clinical point of view regarding this axonal transport, we have to discuss about viruses and toxins. First, we'll discuss about virus. We have these viruses like herpes zoster. For example, herpes zoster, which is a chicken pox causing virus. They cause chicken pox. And they travel from the peripheral nerves. Travel from peripheral nerves through axonal transport, that is retrograde transport, and they will settle in dorsal, that is they will invade dorsal root ganglion. After invading the dorsal root ganglion, they will remain silent for many years, that is they are harbored for many years in dorsal root ganglion. Then what will happen when there is decreased immunity? So whenever immunity is decreased during this harboring stage, they won't cause any disease. But whenever immunity is decreased, then there will be reactivation of virus will occur. This reactivation, what will happen with the reactivation? Virus will be transported along what sensory organs, sensory axons to skin. If they infect skin, what they'll cause? They'll cause blisters. These group of blisters, they in herpes zoster, they are called as shingles. This is how herpes zoster will be harbored for many years, and whenever immunity decreases, they'll cause blisters and reactivation of disease will occur. That is about viruses. And toxins can also be transported, like tetanus toxin. It can enter through dirty wounds. And if person is not vaccinated, if the person has not been properly vaccinated, that is no vaccination schedule for that patient regarding tetanus vaccine, then this toxin will be transported retrogradely to motor neurons. And from motor neurons, they escape into extracellular space of spinal cord. And what they'll do, this tetanus toxin, they will block synaptic receptors. for inhibitory neurons. Inhibitory neurotransmitters. That means there will be continuous contractions and they will cause tetanic convulsions. Tetanic convulsions will occur. This is how tetanus toxin will cause disease in persons when they are infected with tetanus toxin. Now, we'll discuss one more important topic regarding neurons, that is axonal degeneration. What will happen in axonal degeneration? Whenever there is an injury to, like whenever an axon is transected, whenever this axon is transected, what will happen? In a neuron, we have soma. What will happen to soma? That is cell body. Cell body will show chromatolysis. That means ribosomes which are present in the cell body, they appear to be disorganized, and the nisal bodies will stain weakly with dyes. With what dye? Aniline dye. And rough endoplasmic reticulum which is present in the cell body, they will be distended. And the cell body will be swollen. The soma will be swollen and they will become rounded. Normally the shape of this soma is like this and they will become rounded. And nucleus which is at the center position will be occupying eccentric position. That is, nucleus will have eccentric position. Instead of center position, they will occupy peripheral position. That is called as an eccentric position. And there will be increased protein synthesis. And this is a neuron. And this neuron will have myelin sheath. And there is a cut here. There is a cut here. What happens after the cut to the distal part of the neuron? This part, this distal axon to the transection, they will die. And after the death of the distal column, then what will happen to this? Within few days, axons and synaptic endings are disintegrated. That means they are completely taken off. And what happens to the myelin sheath? We have a myelin sheath here. What happens to this myelin sheath? This myelin sheath if it is present, we have myelinated neurons and unmyelinated neurons. If myelin sheath is present, then this myelin sheath will be fragmented and it will be phagocytosed, that it will be digested. So we'll talk about regeneration. Oh, before going into regeneration, all these sequence of events, they are called as these sequence of events which occur after injury. They are called as, this process is called as Wallerian degeneration. Wallerian degeneration. So all the events, chromatolysis, eccentric position of the nucleus, and swollen cell body, increased protein synthesis, and this digestion, disintegration of the distal axon. All these things, sequence of events, come under Wallerian degeneration. And if Schwann cells are viable, then they can undergo cell division. So this is about degeneration. If there is a degeneration, then there will be a scope of regeneration also. We'll see how regeneration will occur both in CNS and PNS, that is central nervous system and peripheral nervous system. So there will be difference between these two systems, that is regeneration, whenever a neuron is damaged. So we'll see the regeneration part. So PNS, that is peripheral nervous system. After axon is lost, that is distal part of axon is lost, then what will happen? These neurons can regenerate new axon. So that means this is a neuron, this is a proximal part, and this is distal part. Distal part will be disintegrated. Okay, only we have a proximal part, and if Schwann cells are present, they will stay. And this is the proximal part. This proximal part will form, they will sprout new sprouts, and these sprouts, they will elongate. And these elongation will occur along the original path of the nerve. That means if the path of nerve is in this area, the axon will also grow in this area only. If this path is intact, then only the sprouts will grow in that path. If that path is not there, it is damaged, then the elongation won't occur. So this is how the proximal part of the neuron will synthesize the sprouts. All these synthesis will occur with the help of neurotropins. These neurotropins will help in synthesis of these sprouts. That is, regeneration is done with the help of neurotropins. And these survived Schwann cells, which are present in the distal stump. So these are survived Schwann cells in distal stump. What is the function of these Schwann cells? They will proliferate and they will form a row like this. They will form a row along the course, and this is a Schwann cells, and they will form a row along the course. And by forming a row along the course, this sprouting axon from the proximal neuron, it will invade these rows of Schwann cells. So they will invade this part, rows of Schwann cells. They invade that, and they will re-innervate the original peripheral target. They'll go up to synaptic area, and they will resume the functions. And these Schwann cells, they will remyelinate axons. And what is the rate of regeneration? It is 1 mm per day. It is a very slow process. And what happens in CNS? That is about PNS, peripheral nervous system. In CNS, neurotropins synthesis is not that much prominent. That's why this regeneration is not done in CNS. That means neurotropins will guide the sprouting, and this guidance is lacking. And so this CNS, there is no proper guidance for sprouts is lacking. If this is lacking, then they can't grow to the periphery and they cannot form any connections. So CNS, if it is damaged, the neurons are damaged, then they are gone. And in CNS, myelination is done with the help of oligodendrocytes. And these oligodendrocytes, they do not form path. They do not form path along which sprouts grow. So this is not seen and there is no further extensions. Everything is lost. So the axonal regeneration is not seen in CNS. If there is sprout synthesis also, then there is no proper guidance and they cannot extend to further because there is no distinct path. So there will be lack of neurotropins influence. This is about axonal degeneration, axonal degenerations. Now we'll discuss one more important topic, that is glia. This is one more cell which is present in the nervous system, and in Greek this glia is called as a glue in Greek. This glial cells, that is glia, they can act as a connective tissue and they help in communication. And what is the speciality of these glial cells? They undergo cell division. In adulthood also, and they can proliferate after brain injury. These glia, based on the size, that is based on the size, they are classified into microglia and macroglia. This microglia and macroglia, this macroglia have oligodendrocytes, they will have Schwann cells, and they have astrocytes. We'll discuss each of these cells in detail. What happens with the microglia? What is the function of these microglial cells? They are derived from erythromyeloid stem cells. That is, they are formed at one place and they are transported to brain, CNS. So erythroid stem cells, and they migrate into CNS early in development. And they have immune response in CNS. That is, whenever CNS is damaged, what will happen if there is a damage? The cellular debris, that is cellular products, are removed by phagocytosis. That is, damaged cellular products are removed with the help of phagocytosis. This is the function of microglia. Next, we have to discuss about astrocytes. These astrocytes are two types. One type is fibrous astrocytes. One more is protoplasmic astrocytes. These fibrous astrocytes are present in white matter, and protoplasmic astrocytes are present in gray matter. And these astrocytes are star shaped. And What is the function of these astrocytes? Okay, before going into function, they will have cell body and they will have several branches like neuron only. This is a star shaped astrocyte and they will have branches. And these branches will give, main branches will give to 100 to thousands of branchlets. What is the function of this neuron astrocytes? They will regulate microenvironment of the CNS. Microenvironment of the CNS. Then, how they will regulate, we'll see. Astrocytes, what they will do, they will contact neurons and they will surround synaptic endings. Okay, they will have a contact with that, and they will also have a contact with capillaries, the blood vessels which are present in the CNS. And after making a contact with the capillaries, and they will form a tight junctions. They will form tight junctions with capillaries. Tight junctions with the capillaries, and they help in formation of blood brain barrier. Blood brain barrier. So these astrocytes, they will make a contact with the neurons, synaptic ends and capillaries, and the these astrocytes, the endings where they are making contact with the capillaries and neurons and synaptic ends, they are called as foot processes. They will be like this. So they are foot processes, and these foot process will make a contact with the capillaries, neurons and synaptic ends. And these astrocytes, they will contact, the foot process will have a contact with pia mater. And through these astrocytes, entry of substances into CNS will occur through this astrocytes. And these astrocytes, what they'll do, they take up potassium and neurotransmitters into them, and they will metabolize and they will biodegrade them and they will recycle. So this is the function of astrocytes, and they will help in maintenance of the micro environment. And what clinical significance these astrocytes have? Any hypoxia condition, in hypoxia, what will happen? there will be ischemia. uh, when this hypoxia will occur, when there is an ischemia due to blockage of artery. That is, whenever there is a blockage of artery, patient will have hypoxia. So blockage of artery will cause hypoxia, and due to this blockage, this is called as a stroke. Due to this stroke, there will be increased potassium levels in extracellular space surrounding a neuron. This increased potassium in the extracellular space will cause movement of potassium into the neuron, and this extra increased potassium will cause depolarization. And this depolarization will cause release of glutamate, and this release of glutamate will further increase potassium release, and this will cause death of a neuron. This will cause death of a neuron. So this will happen. So where does astrocytes come? These astrocytes will take up this extra potassium into them and decreases potassium in extracellular space. With the decrease in potassium in extracellular space will prevent neuronal death. This is the function of astrocytes after injury. So they will minimize inflammation. This is the function of astrocyte. And cytoplasm of astrocyte will have glial filaments, and they will provide mechanical support. This is the function of astrocytes. Next, we'll discuss about oligodendrocytes and Schwann cells. Oligodendrites are for CNS and Schwann cells are PNS. Both will form myelin. So what is the function of this myelin? Myelin increases speed of action potential, conduction of speed of action potential conduction. Okay. And what is the function of this oligodendrocyte? They will myelinate multiple axons in CNS. One oligodendrocyte can myelinate so many axons, and this Schwann cell, they will myelinate only single axon. Single axon. And whenever you take an unmyelinated axon, in CNS, this unmyelinated axons are bare. But in PNS, these unmyelinated axons will have Schwann process. And what is the function of this Schwann process? They will extend to surround parts of several axons. Okay, this is what happens in the unmyelinated axon. CNS, they are completely, the axons are bare, they won't have any cells. But in PNS, they will have Schwann cell process. And this myelin sheath will have proteins, and we have to know about two proteins, that is myelin protein 0. This is called as P0. And a hydrophobic protein, that is called as PMP22. These two proteins are present on the myelin sheath. Whenever there is an autoimmune reaction to these proteins, they will cause a disease called as, and these proteins are damaged with the autoimmune response, and they will cause a disease called as Guillain-Barré syndrome. Barré syndrome. Then patient will have paralysis in Guillain-Barré syndrome. And mutation in the myelin proteins, they will cause peripheral neuropathies. And in multiple sclerosis, there will be damaged myelin, and this decreases conduction, action potential conduction. This is what we see regarding Schwann cells, and if there is any mutation to those cells. So these are the macroglia cells which will participate in the CNS function, that is oligodendrocytes, Schwann cells, astrocytes, and these are the cells which participate in functions of CNS. Next, we'll see some more cells called as satellite cells. Satellite cells. What they'll do? They will encapsulate dorsal roots. And cranial nerve ganglion cells. What is the function of this satellite cells? They will regulate microenvironment. And we have another cells called as ependymal cells. What is a function of these ependymal cells. They will form the epithelium lining of ventricular spaces. So they will form epithelial lining of the ventricular spaces of brain. And CSF is secreted by specialized ependymal cells of choroid plexus, and these are located in ventricular system. So CSF is secreted from the specialized ependymal cells which are present in the choroid plexus. This is about axonal degeneration and glial cells. Thank you.